Tiny brain-like organs grown in the laboratory could speed up the development of drugs for diseases such as Alzheimer's, and even reduce the number of animals needed for medical testing. Scientists successfully grew miniature brains containing multiple different types of human brain cells from human skin cells in the lab. At just 350 micrometers across, they are barely visible to the naked eye and are roughly the size of the eye of a fly, yet were able to send signals much like a full-sized thinking brain. The team say the mini brains could bridge the gap between animal research and cell-based techniques, leading to better treatments and speeding up drug development for diseases such as Alzheimer's or Parkinson's disease. 95% of drugs that look promising when tested in animal models fail once they are tested in humans, at great expense of time and money, explained Professor Thomas Hartung of the Johns Hopkins Bloomberg School of Public Health, who led the research. While rodent models have been useful, we are not 150-pound rats. And even though we are not balls of cells either, you can often get much better information from these balls of cells than from rodents, he added. The brains are grown from human skin cells which were reprogrammed to act like stem cells. Over the course of 2 months, these reprogrammed cells called induced pluripotent stem cells or IPSCs develop into clumps. The resulting mini brain contains a number of different types of adult brain cell, 4 neurons and 2 support cells. One type of cell, the oligodendrocytes, goes on to form the protective covering of nerve cells, myelin, which insulates the cells and helps them to send signals faster. It is this protective myelin coating which is destroyed in multiple sclerosis, which disrupts the ability of nerve cells to communicate with each other effectively. Researchers were able to see the myelin developing in the structures and could see it begin to wrap around the tails of nerve cells, which conduct the signals. The brains even showed electrical activity, which could be recorded with electrodes, similar to an electroencephalogram, EEG, used to detect brain activity. While the miniature organs are far simpler than a complex brain, they provide a sandbox for testing the effect of new drugs. As they can be produced from a patient's own cells, it could potentially be another step towards personalized medicine. A person at risk of a neurodegenerative disease, such as Parkinson's, could have their own mini brains grown from stem cells, with treatments tested on the brains to find which drugs they might respond to best. According to the Johns Hopkins team, the technique can be reproduced on a large scale, generating thousands of copies of in each batch, with 100 of the mini brains fitting into a single petri dish. We don't have the first brain model nor are we claiming to have the best one, said Professor Hartung. But this is the most standardized one. And when testing drugs, it is imperative that the cells being studied are as similar as possible to ensure the most comparable and accurate results. Making the technique reproducible would enable researchers all around the world to use the approach, which has the potential to drastically reduce the use of animals in medical testing. Professor Hartung said, "Only when we can have brain models like this in any lab at any time will we be able to replace animal testing on a large scale." The research will be discussed today at the annual meeting of the American Association for the Advancement of Science in Washington.